So, I want to start by taking a look at this beautiful picture by Henri Rousseau called Tiger in a Tropical Storm. And what we can appreciate here is that we have a very large variety of organismal systems, of biological systems, both in terms of scale and complexity. You can think of bacteria, cells, insects, plants, and the tiger, but they all have the same daily challenge. They all have to survive in a harsh and, most importantly, a fluctuating environment. And it turns out that they do this through a set of, a rather recurring set of complex response processes, such as decision-making, adaptation, and so on. And these are all based on a very similar, on the same ability of the organism, and that is to be able to compare across a history of stimuli, right? It's not just instantaneous responses. And this is what I mean here by organismal memory. And today I want to briefly give two examples of memory phenomena on two very different systems. One has to do with cellular chemotaxis, so we're considering a single white blood cell on the micrometer scale. And the other has to do with plants, which are multicellular systems on the centimeter scale. In both of these systems, and in general, we can identify a motor system and a sensory system. And then there is this very simplistic dogma, if you will, that the environmental stimulus affects the organism through the sensory system. And the end result is a motor response of the whole organism, right? The cell moves, or the tiger moves, or the plant, well, here it's actually a growth response, but still. And in between, there is this black box of various microscopic processes, which are inherently stochastic, right? This is the signal transduction. And the general approach that we're going to take here is kind of rooted in statistical physics, if you will, and that is that given that we know what the stimulus is, we can observe the macroscopic response, and then given that we assume that the underlying processes are stochastic, maybe we can infer something about those microscopic processes. This is the main idea. So I want to start with a problem of directional memory in cellular chemotaxis. This is work done. The nice thing about biology is that you have a lot of movies. This was a very nice change for me. So this is work done with Mahadevan at Harvard, and the experiments were done at the Shah Lab at Harvard Medical School. And here you see these white blood cells performing chemotaxis, so it means that they can sense a gradient of a chemical concentration, and they migrate towards it. Here we have a very, very simplified cartoon of a cell, just to get a feeling of what's going on. We can identify the sensory system as the membrane, which is this, what encloses the cell, and you have your receptors. And inside you have these sticks. Let's say this is the cytoskeleton. This gives the structure of the cell, and also is one of the main components associated with the motor system. So what happens is you have some chemoattractant that arrives, and these molecules bind to the receptors on the membrane, our sensory system. Then some molecules of some type are released right at the beginning. This initiates a signaling cascade, which eventually leads to the localization of the cytoskeleton at the back. And this kind of allows to crunch and move the cell forward in the direction of the stimulus. And it's a very complicated. Sorry?
It's like pulling yourself in your own.
So it's more than that. It has. There are some. There's adhesion and there's some pulling. And I'm really simplifying it here. This is just a representative of the motor system.
There are circles.
Yeah. But something interesting to point is that when you impose an external gradient of the chemical, this imposes an internal symmetry breaking of the cell, both on the molecular level, because these molecules, for example, appear only where you're sensing something, and also on the structural level, because the cytoskeleton goes to the back, right? And luckily enough, in these experiments, we have a fluorescent marker, what you see in green here, which is associated with these molecules here. And indeed, in the movie, you can see that they're really localized at the front where it's sensing something. So, yeah, so this tells us, sorry. So this means that we have two things that we can look at. One is the real external gradient. In this case, it's not only the difference in concentration, it's actually the difference in bound receptors. That's the effective stimulus. But also we have the perceived gradient, right? Just by looking at the localization of this fluorescent marker. So we quantify this with a variable. We call it the polarization state of the membrane. And basically, through image analysis, we just say that this variable goes from -1 if you're completely localized to the left. This means that the cell is perceiving a gradient in that direction. And it goes all the way to +1 if you're completely localized to the right. And this means that the gradient that is being perceived is the other direction. And if it's 0, it means that this molecule is homogeneous and the cell isn't sensing any specific gradient. Very briefly, here you can see a bit better the underlying experimental setup. These cells aren't just free to go, otherwise they would be more blobbish. They go in these narrow microfluidic channels, and you can see that here we have two reservoirs where we can tune the concentration of a chemoattractant, right? We have C1 and C2. And the nice thing is that these channels are narrow enough so that they constrain the cells, and then they act as plugs. So no liquid can pass from one side to the other. So this means that I know exactly what the cell is sensing on either side. This is our delta N, basically, the external gradient. Giving us basically a one-dimensional problem, which is a miracle in biology. So this is very nice to work with. Okay, so now I want to define a directional memory within the context of this experiment. So we look at the case when there is no gradient, right? The two concentrations are equal in both reservoirs. And in this case, cells tend to spontaneously polarize and depolarize due to internal noise. This is a lot of hand waving right now. So we look at a polarized cell, we wait for it to depolarize, and when it repolarizes again, we ask ourselves whether it repolarized in the same direction as before or the opposite direction. So in the case of 0 nanomolars, meaning that there is no chemoattractant whatsoever in the system, you have roughly 50% of the repolarization events are in the same direction. Okay? So this is what we would expect from random dynamics, right? There's a 50-50% probability of repolarizing the same direction as before or the opposite. But something interesting happens when we look at a non-zero concentration. So there is still no gradient. This is important to note. And both reservoirs are now with 10 nanomolars. And now you see that roughly 80% of the repolarization events are in the same direction. So this is what we mean when we say directional memory. The cell seems to remember the direction that it sensed earlier on. Now you can also take this one step further and try to probe the time scale of this memory. And we can look at repolarization events that happened after 2 minutes and after 10 minutes. So you see that there is this slow relaxation of this memory effect. And after 10 minutes, you still have about 70% of the repolarization events in the same direction. So this means that the time scale of this memory is on the order of tens of minutes, which is much longer than the time scale of seconds, which is on the order of the sensing system, sorry, which is in the order of seconds. Yeah.
What's the waiting time for the first one? For the 10
This is just everything. We average over all the repolarization events regardless of the time. Here we just filter out repolarization events at a given time, or you can also force it to repolarize by removing chemoattractant and introducing the chemoattractant. So there are two ways of getting a repolarization at a given time. But this is averaged. So you can say that this is an average of these two, if you will.
Do you have an optimal time for memory? Like, do you have an increase of the memory and then decrease over time? Or?
Well, the shorter the time, the higher the memory. And since these experiments are complicated, we only have here 2 time points. Here, this is just to probe the time scale in general. The point here is, though, that since we see that the time scale is so much longer than just the sensory system, means that this is not enough, right? We can't just look at the sensory system, which was what has usually been looked at before. And it means that we probably have to also introduce the motor system somehow. But before we do so, we first have to provide ourselves a mathematical framework that describes the dynamics that we see here of these cells, so that we can actually do something interesting. So we're going to look at 3 general cases of types of dynamics, and from this try to build a description. So this is a sanity check. You have an example here of a single cell where there is no chemoattractant, 0, 0 both sides. And remember that we have the polarization state, PM, the polarization state of the membrane, how localized this fluorescent marker is as a function of time. We have many such trajectories. So now I can chop up these trajectories and create a distribution, a probability distribution of this variable. And we see that, as expected, we have 2 symmetrical peaks around the polarized states, +1 and -1, right and left, and a smaller peak around the unpolarized state. Another case is when there is a very big difference. Here you have a saturating 100 nanomolars on one side, and you see that the cell just shoots in the right direction, right? So you have a huge peak at +1, no mistakes, never goes in the wrong direction, and no depolarizations. And lastly, something in between. Here you have a small concentration, and now fluctuations, both within the system and due to the receptors, the binding and unbinding of the receptors. So now you see that he's not so convinced that this is the right direction, right? So you still have the bigger peak around the correct direction, but it makes mistakes, and it does unpolarize. So the point being, now that we know that we have stochastic dynamics, we see that there are preferred states. This suggests, hey, maybe we can use a Langevin equation with some kind of effective potential. This is, of course, not a real potential. So what we're going to do here is make an educated guess. We choose a double well potential, so that in one, you see here that we have the prefactor alpha, which is the sensitivity, basically, since we're looking at the sensory system. So in this case, the stable states are the polarized states, and you can go between right and left, or +1 and -1. If you now flip the sign, the stable state is the unpolarized state. And I can also add delta n, which is the external gradient, right? And this will give me a slope, and then there will be a preferred state. And what we do is, so here is our Langevin equation. This is our double well potential. This is the gradient, delta n, and it is the noise. And now we allow alpha, our prefactor, in itself to be a random variable. And this will allow it basically to change, to switch signs randomly. So this is a more exotic Langevin equation, let's say.
This is without memory.
This is all without memory. This is just to provide a framework. And indeed, this brings me to the next slide. This, for now, I can tell that numerical simulations indeed reproduce all of this. But as I said before, in order to get memory, it seems that just looking at the polarization state of the membrane, which is a sensory system, isn't enough. So now we want to consider also the motor system, the dynamics of the cytoskeleton, which follows the same idea. So this is what I just showed now, the equation for the membrane. Now I can write an analogous equation for the cytoskeleton, again a Langevin equation with the same double well potential, but now the bias is not delta n. Delta n is from the environment, right? But the cytoskeleton is inside the cell. It doesn't know anything about the external world. All the information comes from the membrane. So hence the bias is given by the polarization state of the membrane. But we have signal transduction in between, right? So information isn't instantaneous. It trickles down. Some of the information arrives now, some of the information arrives later. It's a stochastic process. So what we do is we convolute our polarization state of the membrane with this memory kernel or response function, if you will, which encapsulates the stochasticity of the signal transduction. And this is also what tunes in our time scale, the long time scale of the motor response. And now there is one last ingredient that we have to add, because this is not enough to reproduce what we see here. There's a lot of experimental evidence that shows that the cytoskeleton actually affects what is being sensed on the membrane. So the last thing that we add now is a coupling also upstream, not just downstream. So now the bias for the membrane is not only due to the environment, delta N, but also due to the state of the cytoskeleton. So now we have two competing biases, because you have to remember that this is instantaneous, this is from the external world, but this has a lag, right? So now we have this interesting coupling. And apart from the fact that the numerics reproduce the observations of directional memory, the important thing is that we can make a prediction here. We can say that we see from this we understand that the memory is due to this coupling between the cytoskeleton and the membrane, between the slow response of the motor system and the fast sensing. And we can verify this experimentally. So indeed, this is the control experiment that I showed before when you had 10 nanomoles on either side and we saw directional memory for the first time. And these are analogous experiments, but with various drugs. And these drugs affect a protein called mucin, which is this one here, which basically connects the cytoskeleton to the membrane. So these two drugs decouple mucin from the cytoskeleton, or they mislocalize mucin, so they do something bad. And you see that memory goes almost to zero. On the other hand, this drug stabilizes mucin, and you see that memory shoots up. So this is really a nice verification of this prediction. I should say that these drugs do not affect the ability of the cell to sense or to migrate. And so now we can wrap this up, saying that we have a model that describes, first of all, it's interesting to note that it describes a decision-making problem, first of all. And it was able to reproduce the memory effects that we see and make a prediction. And we found that this prediction is due to this coupling. But now you can ask yourself, since it's important for a biological system to be robust to fluctuations in external stimuli, right? Maybe this could be a useful general strategy in simple biological organisms. And this brings me to the next very short part of this talk, where we look at the ability of plants to integrate stimuli over time. So this is a very different system. Again, it's multicellular, it's on the centimeter scale, but we're going to see that there are very similar elements here. So first of all, I want to define tropisms. Tropisms are the growth-driven response of a plant to an external stimulus. It can be light. This is phototropism. This is when you put a plant next to a window, and you'll see that it senses that the direction of light comes from here, and it will grow towards it. This is the more known one. And another one is this one, gravitropism. Notice the date of this movie. This is from 1898. I find this pretty cool. And this is basically that if you put a plant horizontally, it senses the direction of gravity, and it grows in the direction. And this is the shoot. The roots go down. So it's a very useful property to have for a plant. And here you see snapshots of a wheat seedling placed horizontally after a couple of hours. And this is just to point out that this, as I said here, this is not moving, right? This is not bending. This is growth-driven, right? So it means that you have asymmetrical growth. And this is mediated through the redistribution of a growth hormone. So here you can see, this is an inset of this part. The yellow dots, which represent this growth hormone called auxin, are homogeneous. And this is why the shoot was still growing straight. But as time goes by, as it senses the direction of gravity, we see that there is a redistribution of auxin only to one side, and that side grows more quickly than the other, and you get this bending. Okay, so the interesting thing is that more than 100 years ago, by now, there have been observations showing that plants do not respond instantaneously, but rather to a history of stimuli. And particularly, there are two classes of experiments. and this is both for phototropism and gravitropism, so completely different sensory systems. Reciprocity is, for example, these are examples of stimuli. So you have here a stimulus with a given intensity and a given duration. And here you have a different stimulus with, let's say, half the intensity and double the duration. So it's a reciprocal ratio, but the dose is the same. And it turns out that plants respond exactly the same to these two stimuli as long as the dose is the same, within a certain window of time, of course. A second class is summation of consecutive stimuli. So if you have these two time-separated stimuli, the plant again will respond identically to this or to the continuous version of these two pulses. Okay? And the question is, why is this happening? Where does this come from? So very briefly, we use as a mathematical framework this very nice work by Bastien Beau, Moulian Douadi, where our variable here is theta, the local angle. So this is our plant shoot, and S goes from the base at 0 to the tip of the shoot at L. And now we can, basically this is the angle from the vertical at point S at time t. And I've rewritten this equation in a way where you can separate, on the right-hand side you have the affecting stimulus. So theta p is the angle of the stimulus, and we're looking here at the difference between the local angle and the angle of the stimulus. The idea, the goal of the plant is to minimize this angle. Beta here expresses the magnitude of the stimulus, the intensity. And here is a growth-driven response. I won't go into the details to explain this, but you can appreciate that that it has to do with growth because you have all these derivatives in space. And what we did now, this is completely instantaneous, so by definition it cannot explain what we just saw. But what we do now is something similar in spirit to what I showed with the chemotaxis. And I want to, the point is that what's missing here is a signal transduction, right? You have the stimulus and you have the end result, but you don't have the black box in between, right? You don't have the redistribution of auxin, for example. So we encapsulate in this response function, and we now allow also beta, our stimulus, to be a function of time, not just a constant as was done until now. And again, numerical simulations managed to reproduce the experiments that I showed in summation and reciprocity. But we can take this, and I'm almost done here, one step further and say, can we connect this to something experimental, something with a biological relevance and not just a nice phenomenological description? And the answer is yes. Since this is a linear response function, by definition, if we give the response to a pulse stimulus, will be the response function. So if we now substitute beta with a delta function and fiddle around with the equation, we can represent our linear response function mu as a function of trajectories of the angle of the tip and various other parameters which are all experimentally tractable. And as we speak, we're running experiments in the Moulia lab in France of pulses of gravity. And hopefully we'll be able to say something interesting very soon about this. And the idea is to associate this, of course, with the stochastic transport of auxin. So that's it, I guess. Thank you very much. Yeah.
How is it actually sensing the gravity? Is like an enzyme that's suddenly at the bottom of the cell, it's spitting out oxygen?
It's actually the initial part of the sensory system is quite mechanical. You have a big cell and you have a myeloplast, which are organelles with starch. They're just heavy, and they sediment. That's it. The problem is that not much is known in the sense of what happens when they sediment. Then there is some kind of signaling cascade which is not well understood. But the basic idea is that. Yeah.
And the time scale for sedimentation, or in the case of the cytoskeleton, what would be the time scale for the collapse of the cytoskeleton? Be the myosin motor or something?
Are you saying that?
What sets the time scale for both processes?
Okay, so in plants, the sedimentation is roughly 3 minutes, but the response, so in fact with the pulses that I was mentioning, the idea is that you have to try a stimulus and shorten it and shorten it and shorten it until you see what's the minimal duration of a stimulus and that you still see a response. So 3 minutes will be our lower end, and right now, surprisingly, it seems that that's also the order of magnitude that you need to see a response, which is surprising because we assumed it would be longer than that. Within the cytoskeleton, it's on the order of maybe half a minute, but this is a bit, it's a bit difficult to answer that because we're not looking at the cytoskeleton per se, we're looking at it as a representative of a larger picture.
Are
Yeah.
there long-term memory effects as well? So that put it in a polarized condition for 1 minute versus 1 hour, and then measure how long it takes for that memory to go away?
That's a good question. We haven't checked that. But it is a matter of what I didn't show here, that the motivation for this was an experiment called a wave paradox, where it's a different type of cells, but still, you put cells and then there is a moving wave of a chemoattractant. So whilst the wave is coming towards you, the direction of the gradient is that way. So you see cells migrating towards it. And when the wave passes you, you expect the cells to switch directions and follow the moving gradient. And it turns out that there is a region of velocities of this moving wave where they seem not to notice it and they continue straight. And this is basically this can be explained due to this competition between the timescales of the external change in the gradient, delta n, and the bias of PC, the cytoskeleton, which doesn't keep up.
Okay, let's thank